coming to this symposium. I have a pleasure to meet during these days a group of very good scientists working in different areas related with the phenotypic plasticity of marine organisms when changing in the ocean. So we are going to talk this morning, about by using different groups, as models for trying to understand what is the impact of the phenotypic plasticity on marine organisms. This is a part of a project. It's a network project funded by CONICET. So with this group of scientists, we are trying to implement a global experiment which is going to be carried out in different parts of the world to try and understand which is the response of mussels, in this case, as a model under changing conditions in the ocean. So just a brief introduction. So this is working. So well, it's pretty well known that there are many changes that are occurring actually now in the ocean. Most of them related to the increasing of carbon dioxide concentrations in the atmosphere, which is causing changes in the oceans in different environmental variables like increasing temperature, the reductions of the pH in the ocean as a result of the increasing carbon dioxide both in the atmosphere but in the oceans, and the reduction of the oxygen in some areas. But all those stressors are interacting in the multiple ways, and the impact of those stressors is different depending on the different populations that we can find and along the coast of the different continents in the world. So global change in the ocean is already affecting the physiology of the marine organism, and of course we need to try to understand which is the response of those different groups, these environmental gradients, which is the environmental limits for different physiological traits, like growth, like reproduction, like survival. So, well, the impact of different multiple stressors like ocean acidification, global warming, is not only affecting, individuals, populations, and communities, but also is impacting different ecosystem properties, the functions, and the ecosystem services in different areas. So this is a picture of a recent paper published by Howell, Spencer, and Harvey, where they show how, by studying environments with a very high concentration of carbon dioxide, they have found that the loss of habitat forming organisms and degradation of ecosystem is extremely important because it's affecting, the different ecosystem services that can provide some specific areas. And some of the impacts are related, with the reductions in the habitat complexity, the species richness, and especially the habitat coverage. So these impair ecosystem functions in some ways, and of course the goods and services available to society, such as coastal protection, the recreations, and the food provision. So one of the important questions, for the scientific community is trying to understand what is the adaptive capacity of the different marine organisms in the face of those changes in this change in oceans. But in case that some organisms have the capacity to adapt to those changes, so obviously next question is try to understand, okay, so how far can they go? So the study of the phenotypic plasticity is a good way to try to understand, which is the adaptation that some organisms can develop to try to deal with all these kind of stressors. So phenotypic plasticity can be defined as the ability of a genotype to alter its resultant phenotype in response to an environmental change, like temperature, like pH and oxygen. This is a classical picture, that show, for instance, two genetically identical water fleets. It's a Daphnia. So, and the helmet and extended tail spin in this individual, for instance, on the left, is a result, of the chemical cues produced from a predaceous fish and serves as a protection for this one. So, in this case, it's a result of this plasticity. So the particular way of that individuals or genotypes vary across environments can be described like reaction drops. So you have a gradient of a specific environmental variable like temperature or pH, and you study a specific phenotypic trait, and you can have a curve that can define how plastic is an organism. So in this case, for instance, you have organism with no plasticity, so there's a gradient in the environment, and the trait is there's no impact in the specific trait. We have seen in this case that some plasticity, and in some occasions, cases of non-parallel reaction norms indicate genotype by environment interaction, which is underlying the variation in organismal response to a fluctuating environment. Is a kind of regulatory genetic architecture responding to selection in this case. So actually now we can, try to define that there are different kind of plasticity. Is the adaptive and non-adaptive plasticity. So Juan Diego and Sam, especially Sam, is going to talk at the end of the session about the difference between both. You have to recognize that plasticity may be adaptive if it's maintained by natural selection, or non-adaptive, depending on this relation with the optimal fitness in the new environment. In the case of non-adaptive plasticity, this produces phenotypes that are less fit in the new environment, so it's extremely important. This is just a scheme that I did, to try to represent, the difference between adaptive and non-adaptive plasticity. We can see that this typically arises when the costs associated with plasticity, outweigh the potential benefits. You have two different traits like growth and reproduction, for instance. So there's a kind of cost-benefit relationship. It can also purely indirectly if selection results in an energetic, for instance, trade-off between, traits and plasticity. For instance, there's a positive chain in one trait, like, calcification or ingestion rate, but it's compensated by a negative chain in another one, like reproduction. So it's extremely important in order to try to have a more comprehensive understanding about the plasticity of the different marine groups, to try to understand the response or the performance curve or reaction curve for different traits. So in the literature, you can find that there are different, definition of phenotypic plasticity, but also other associated terms, and more specific terminology like, canalization, developmental instability, phenotypic flexibility, developmental plasticity. But regardless of the subfield, phenotypic plasticity is an important concept in modern ecological, evolutionary, and biomedical literature, and it has been a primary interest for different research groups in the world, especially in the framework of the changing ocean conditions. So regardless of the nomenclature, physiological adjustments in response to environmental heterogeneity are generally considered to be adaptive and advantageous. So during the last 4 or 5 years, there's an increasing amount of information that you can find in the literature trying to understand, the phenotypic plasticity of marine organisms under, global chain by using different kind of models. And as you can see that during the last 10 years, it has been a huge increasing of the papers published, especially focused on the study of trying to understand the phenotypic plastic response of organisms of the changing ocean. And the models that the community has been using is very wide. I mean, this is just an example, for instance, of the impact of a two-point reaction now to try and understand the plasticity of 3 different groups of phytoplankton. This is Chaetoceros affinis and 2 Coccolithophores, Emiliana huxleyi and Hydrocapsa oceanica, 2 different concentrations of CO2. And this paper by Hatch, they were looking, about the plastic response of those different groups in the row rate. So by using as main drivers the CO2. But in the other case, you can find, for instance, other kind of model like fish. And in this case, in this paper by Bowman and Conover, they try to understand how fish population, in this case, by looking at the growth as a main trait, to try to understand which is the effect of the temperature, by considering global warming as a major processes, and how increasing temperature can affect the plasticity in this case of fish. But of course, there are different issues that needs to be improved, especially in the case of planktonic organisms. So there are a lot of discussion, for instance, for phytoplankton, it would still needs to reevaluate the correct traits for evaluate plasticity. This is something that we have been discussed with some people. So what about in Chile about this issue? So we have been with a group of colleagues from different institutions between 2009 2014. We have been trying we have been conducting different common garden experiments to try to understand the plastic response of different local populations along the Chilean coast, by using gastropods and mussels as a model. So basically we have been, collecting animals from different populations like in Antofagasta or in Las Cruces or the southern part of Chile, like Valdivia or Chiloé, and then we have moved into the same laboratory and exposed it to the same conditions like food, temperature, and different, pH level to try to understand, how plastic are different populations along the coast of the changes in the pH and the carbon dioxide concentration. This is very useful information because it also can serve, to try to understand the resilience of different populations along the coast of global change conditions. So, which can be very useful not only for the community, for the scientific community, but also for stakeholders interested, in marine reserves, or especially for the aquaculture, which is carried out along the coast of Chile. So many papers were published during that period, with different experiments with gastropod and mussels, but I have, with my colleague, we have tried to synthesize that information in this paper that we have recently published in Nature in 2017. So, and we were trying to understand how we can explain, the different response of all those different populations by considering the specific variability in the pH in the respective area. So by using information from sensors, from time series, from different, monitoring programs, we have been trying, to have an idea about how is the natural variability of carbon dioxide and pH in all those different sites along the coast of Chile. And so by using this information and putting, all these experiments in the context of the variability of this local population, we have found very interesting results. This is a kind of a standardized, response. For different traits in different organisms like copepods, gastropods, and mussels, based on experiments in the laboratory. And this is a kind of index that represents, how far the organisms collected in the respective areas are from the comfort zone or the extreme conditions that can experience in the respective habitat. And this very nice relationship between both represent that organisms exposed to large changes in PCO2 from the extreme environmental conditions are the ones evident the largest negative mean effects. So it may suggest for the Chilean coast that there is a kind of local adaptation to a very high PCO2 conditions, might lead to an adaptive response, resulting in a reduced negative impact in the study physiological trait. So this is a very, very useful information because it may have some implications in the selection, for example, of genotypes more resistant to high PCO2 conditions along the coast of Chile. So, well, the understanding of natural variability leading, plastic response is very important because you can use this information for validated experiment with species from different environments, like coral reefs, rocky shores, alkaline areas, estuaries, and fjords, and then try to understand why, different populations respond in a different way to changes in the environment, not only pH, but also, temperature, or oxygen, or different stressors that you are interested in studying. So local variability, the other aspect, is that shape different phenotypic traits in a different way. For instance, this is a kind, a plot for different processes occurring at different time scales, from tides to diurnal tide, storms, upwelling, to fluctuation associated with a process occurring a very long period of time, like geological changes or the increase in anthropogenic CO2. And you can see that processes occurring at shorter time scales can produce significant variability, but smaller perturbation to the long time scale driver who may have a higher effects on higher levels of biological organization, like evolution or at the level of population or communities. So it is suggested that adaptation potential and resilience are found at higher level of biological organization whereas acclimatization may occur on the lower levels of biological organization, like the shell formation or the larval period. So basically, we were trying to understand the last time, and especially now in the framework of this project, is trying to have an idea about how the natural variability in different environments can induce different plastic response or performance curve for one model, biological model. In this case, we're focusing in MUSELS, in a cosmopolitan species that you can find in different areas like Mediterranean and the coast US and Mexico and Chile and the Indo-Pacific, and trying to understand how natural variability can make a significant impact on the response of those organisms to changing or the future changing ocean conditions. So okay, so today we are going to talk about different issues related with the plastic response of different populations. So Bernardo, for instance, he's going to talk about the environmental variability in the coastal zone. Eugenio is going to talk about phenotypic plasticity in sea urchin and mussels in the California coral system. And then Marco is going to use as a model the gastropod species. Juan Diego is going to talk about phenotypic plasticity and adaptive evolution. And finally, Sam is going to talk about adaptive or non-adaptive plasticity, or what is important to make the difference. So thank you very much again for coming, and I would like to, after this introduction, to present the first speaker of this symposium. Is Bernardo Broitman. He's a researcher from the Universidad Adolfo in Viña del Mar, and he's also the deputy director of our MUSELS centers here at the Universidad Concepción. You have your pen there?
Patience, keeping on the edge.
No, you can. No. Here?
Here.
Okay.
Starting my own presentation. We should do that randomized and it should be the other.
Bueno, buenos días, good morning. Nos dijeron que hiciéramos esto en inglés porque tenemos algunos angloparlantes y tengo casi todas las diapos en inglés también, así que no les voy a poder ayudar mucho por ahí. Pero, so this is like fully in English because I have no slides in Spanish, which I thought I would, but I'm going to keep it down. So I'll be talking mostly about the intertidal zone, but I put some data from very near shore oceanographic moorings that you'll hear more about them later. And through this data, the idea is that I'm kind of like the introduction to what's next in the morning. So I'll be showing you what are these like dominant temporal scales in environmental variability. So basically, how we have characterized the environment that the organisms are exposed to, which is what it will inform most of the experiments that we have been doing and that we want to do later. I'm going to blend that kind of like temporal aspect with the space-time variability, so how the temporal changes are different through space as we move through geography, and then maybe try to discuss what have we learned with this. Okay? So I'm going to get started. Everything that we've been talking as of late and we're in Concepción is from the fjord area, which is most of where the aquaculture is concentrated. but I've been doing a lot of work in the desert coast of northern Chile. This is Taltal, just south of Antofagasta. This is where the extreme desert begins. And I worked for a long time in the Center for Arid Zones, and people told me, like, if you're a marine person, why you work in the arid zone? Because, well, arid zones are characteristic because they have strong coastal upwelling, which is why they're so dry. So this is where Taltal is located. Antofagasta is right here, if you might know where Antofagasta is. Coquimbo is right here. Santiago, Concepción is down below here. Chiloé down there. So Chile is a very long country. We are all Chilean folk are told in school. But when you look at like global data, Chile is very interesting because Chile in latitude runs straight up and down. So latitude moves very quickly as you move along the coast. If you go in California, it will take you a very long line of coast to move through so much latitude. Okay, so in this space-time variability, Chile is a very good study system because you can learn a lot moving not too much. Well, this is a long gradient. So this is what Taltal looks like, and Paposo and Taltal are interesting because they're quite contrasting. So you can have very persistent changes in temperature regime and in some other oceanographic aspect, or a very short stretch of coast. And this is part of what we have been learning over the last years, is that the ocean is very well structured, much more than we're used to think, because oceanography is always told kind of like way out there, where it's very stable, but the coast is more abrupt the way it changes. So part of like trying to understand how the ocean is changing and the scales of variability in the ocean need for us to think about longer time scales. And the longest time scale that you can go to, if you have the money and you're in the correct place, would be to use a paleoceanographic approach, because you can get this very long course of sediment that can teach you what happened in the past. The best we can do here is work with the archaeology, so do some interdisciplinary work and examine the stable isotope composition of seashells. So we've been working with a group of archaeologists that they work in the Paposo area. This is, you can't see that, but this is full of shell mills, which is where people lived before. So you can get the very good sequences of shells and date the shells directly, date where the shell came from, and get a little look at what was happening on the shoreline oceanographic. Okay, so there you can see like the hole that you drill to get the carbonate from the shell, and then like the little vial that you send for analysis. And it was published last year by Carola Flores. So this is the latest. We are in this paper. We had like half of this data, so now we need to publish this pretty soon. And what you're seeing here, this is very interesting, and it's not being looked at in Chile as far as I know. So this is the full Holocene in the temperature using isotopes, so stabilized isotope, and the C14 of carbon, which tells us something that we need to be careful the way we interpret it, but it tells us something about the intensity of upwelling. Again, I'm going to be cautious about this one because you need to validate using more proxy, but it will give you like a rough look at how the intensity of upwelling was in the past. So look at the number. So we have like 12 degrees Celsius, 18 degrees Celsius in the oxygen isotope coming from the isotope. And what we can see is that the early Holocene, when glaciation was finishing, the ocean was cold in Taltal, 25 degrees north. Then in the middle Holocene, we had the warm period, the climatic optima, called by the archaeologists, because it was really warm and moist. And then it cooled down again. Around this time is where people think that the actual ENSO regime, the El Nino regime, got started. It cooled down, and then it started warming again. And then this like zero data are our data from those shells that I'm showing here, from this like modern shelf, and some data that it was published earlier by some other authors using different shells but using carbonate. And you can see that it's getting warm. We're warmer than in the mid-Holocene, or just as warm. But the upwelling, it seems to be, or the signal of upwelling seems to be pretty intense in this shell, which means that we're not getting the data correctly, but it kind of makes sense because the early Holocene seems to be very cold. The upwelling system in the Humboldt current as we know it, it goes back kind of around that day. So this is a short lesson from the interdisciplinary collaboration. So there were these important oceanographic changes during the Holocene. We knew that already. However, there's very little data in between the tropics, where people went with coral, and in the far south, where they have done more oceanographic work. We see the warming. We see that it's getting warm now. I'm not talking about climate change, but currently, 20th century maybe, is warm. It's like the mid-Holocene period, and there seems to be this intensification of upwelling. And I didn't show the data, but there are no across the whole period. The ecological composition, the species that live there, they have not changed a lot. We have some data showing that from the shell video, but what we're working on to see if the ecology of the place has shifted, like the relative abundances of species have changed. So now back to the present. This is the rock intertidal zone in central Chile around Los Molles. And what you can see here, those are mussels, that people are like running away from the wave. These are kelp beds, and this is kelp that had been burned by the sun during the summer. So if some of you, because I know there are all kinds of students here, don't know the intertidal zone is very interesting because you can be sort of in the ocean, but you can get also this strong atmospheric signal as well. And how do we study intertidal zone, the environment, the high-frequency environmental variability? Well, we'll try to keep it simple because it's a not very easy place to work in. So we work with the temperature data logger that you can get a multiparametric sensor to, and we attach them to rocks or we tie them to pilings, but they're just underneath the water. So the environmental signal that I'll be talking about, it will be scattered with different things. It's very near shore. So what organisms are expecting there. So if you use these little data loggers that are now not put into the PVC pipe, but they're into a concrete block that is like sunk in the shore, kind of like a meter below low tide. So it registers what's happening in the water. You can work for a long time. You keep changing them. They're disposal because they drift, so you throw them away and put a new one. But you can keep your data going for a long period of time. This data, it's in a complicated digital format, so we'll get there at some point. But we have this 20-year record of temperature between Cabezal Bajo, which is south of Taltal, because it was way too complicated in 97 to work there, but we could get safely, got started to the which is right around Coquimbo, here in this big bay. Why we were interested in that? Because back in 97, we knew that these mussel beds that I'm showing here, which are a characteristic feature of the ecology of the mid-intertidal zone of the rocky shore, they disappear north of Los Molles. They're not there, and we suspected that something was going on in the oceanography. So we started in 97 the monitoring program, and well, the northern region was warmer, the southern region was colder. Here, Valparaiso is here. So you get kind of the idea where it is. So it's colder in the south, warmer in the north, but it's not also that it's colder in the south, warmer in the north, which you would expect, but it also is very abrupt where it changes. Okay, you see this like very clear line. This place is called Punta Lengua de Vaca. It's a well-known upwelling center, and well, Fabian is here and some other people that got started back then, but in 1997 we were not so sure because satellite images, for example, were just coming out. So you could see that there were some things going on, but we were not sure what. So it was not only hot and cold, but there was some difference in the dynamics, which is what I'll be showing later. Also, now that we have more data, if you remove the annual cycle, you see like this like banding pattern, those are the years, summer, winter. You can tell that the large perturbations are very clear in the data. So this is the 2015-16 El Niño, this is the 97-98 El Niño, the 2007-8 La Niña. So these little data loggers that it took us a long time to convince the oceanographers that we were actually studying the ocean, they are actually doing quite a good job measuring what the ocean is doing on the coast for the organism. Okay, and I was saying that we have a change of regime here, a regime that is not so apparent when you look at like hot and cold. When you have more data and can look into the climatology, so you have fall, winter, spring, summer, you can see that as you move south of this bay, the dynamics change completely. You have this very long and wide annual cycle inside the bay and up north, and as you move into the upwelling region, the range of variation is much smaller, and it's quite noisy, but it's fluctuating on a narrower range, which is the signal of upwelling. It's also a lot colder. If you remember the data from all across the Holocene, it was fluctuating exactly around this scale, 18 to 12 degrees. So this is important to keep in mind from the science side, because you can use a smart choice of sites across geography to represent a lot of variation, even like the variation you will get through the entire Holocene. Where I'm not saying that this is the Holocene, but you can use it as an experimental system. So when you look at like now at the whole country, again, these are the climatology for temperature, for chlorophyll, and for turbidity. Okay, and this is using now we're in the present, correct? Like we got time 97, now we're in 2017, 20 years later. So we have the MODI satellite. It's been now for I think like 18 years going around. So you can make good climatology. You can rely that you're capturing correctly the annual cycle. So what do you see in the temperature? Nothing what we knew before. There's a large scale gradient in temperature. The south most of the time cold, and around here you start seeing the very broad annual cycle, which is what we were capturing in the image before. But also that the chlorophyll is really well spatially structured. So you have those peaks, for example, there. That's the Antofagasta Bay. So those are like kind of individual upwelling center, but we're not sure why they're related to turbidity, which they might be related, that it's been described to as dust plumes. So the environmental signal that you can capture, it gets more, there's a word in English called nuance, that que tiene más matices, as you get more data, because it's not only that the chlorophyll is high, but also it seems that some other driver is playing into that. And turbidity in the south is very clear, so all these stripes that you see here are the mouths of the big river. This is the Biobío. This is, sorry, the Maule River. Biobío is down here. You see the plume of the Maule is huge. And as you move further south, you see like individual drainage spike out during the winter months, which is the season with maximal flow. And here that turbidity is showing up during the season of maximal wind. Correct? And here the chlorophyll, the peak in the chlorophyll doesn't. It's not that when we're looking at the ocean color, we're looking at turbidity in the water, but it might be driven by some other colored stuff that is in the water, correct? So we have been going from the Holocene through the Rocky Shore on a small region, and now the whole country, trying to capture like what the organisms are experiencing. But you need to look a little bit deeper, correct? We're not looking that deeper because we're like in this sensor. This is a pH meter, a CFET. This is a CTD, a water quality monitoring, and there are more, one in Chiloé and one in Tumboya. In Chiloé there are two. But these are they are more in the areas where aquaculture takes place. In Tongoy here up north, we have the scallop aquaculture, and then down here in Chiloé, we have the mussel aquaculture. So part of one of the MUSELS project we've been doing is that we're trying to characterize environmental variability where the productive activities take place from a scientific interest, like driven through questions. We're not trying to like maximize production, but we're trying to understand what is driving an important socio-ecological system, as the case of aquaculture. So the time series that we've been getting in the north, the data is a little bit longer because we were working with the arid zone center there with CEASA. So we have this mooring station that it was online for a while because the pole broke and fell in the water. Those are the vagaries of monitoring the ocean. And just like look at the time series, that is very, very noisy, kind of like shifty, going up and down. The oxygen is very low. It's really in the surface, but the bottom oxygen is like really, really low, particularly during the La Nina, after the El Nino of 2015. And salinity is kind of like flowing on a different pace. Don't go. And when you go south to Chiloé, we have a shorter time series. It's longer now. One of the moorings got changed. But what you can see is that the fluctuations are not so intense as they are in Chiloé, sorry, in Tongoy. And also that the annual cycle is kind of smoother, right? Here it's like 16 to 12, and here, look, it's like 20 to 10. So it's kind of like moving around more at a different pace. But that's not the way you look at time series. You need to look at the structure of the time series. And we published a paper earlier looking at this particular time series and the frequency component. And now Luisa is preparing a paper with this data looking at the more finer scale, because we're interested into what the organisms are experiencing. And actually, what do you see here is not terribly clear at this point because the time series for Chiloé are not long enough already, but they are, we need a little bit more data, what scientists, we always have to say, right? But we have very clear daily and semi-daily period in both in the north and the south, which are related to the fact that in Chile we have symmetrical tides. So you see like twice daily peak in all the variables that we looked at. And we're seeing some stronger variability around like longer time scale, probably related to the tide, to like the full tidal cycle, but not so clear anymore. So the lesson we're getting from this is that probably an important signal over this length of a data set will be the dial cycle. But the dial cycle really is what the organisms are experiencing all the time, correct? Day and night, it happens for sure. But the tide, the intensity of the tide, maybe not so much. The other thing is that this is not the right scale, but the data in the north, it seems to be more energetic in total, like it fluctuates more, or this like shorter time scale. So semi-daily and daily variability are important, clearly. The day and the night will happen for sure. And we have this question of how important is the monthly cycle. I mean, the monthly cycle, okay? But the lesson we get from documenting environmental variability is that we have these predictable scales of variation across the region that we were looking at, okay? Which is something we need to keep in mind, and we need to look a little bit deeper into the data once we get longer data. Because, for example, you have 2 years, you cannot look at the annual cycle because you have 0 degrees of, you have 1 degree of freedom. So you cannot compare 2 years. So if you have 3, better. You have 5, much better, correct? So we're getting there. It's a slow process. So the other important source of variation that is kind of in between all of this is El Nino. Okay, in Chile, we're very used to think about El Nino, but when you're like in Hong Kong or in Denmark or in Sweden, you're not thinking about El Nino. But here, like El Nino is very, very present in our life. So this is what the 2015 Niño looked like, and this Niño was very weird because the northern hemisphere was warm already before the El Niño, and then it got even warmer. And that way, Chile didn't get so warm. It did warm. Like I showed you the data, it was a warm year, but it wasn't a particularly strong one. So when you look at the data, what I was showing before, you see that, yeah, we saw El Niño here, but the anomaly back then in 97, we don't have that much data, but the anomalies were much larger. However, when you look at all this data together, and you calculate the regional anomaly, you tend to see that we have the bias for lacking this piece of data set, but we know that across the region examined, kind of the long-term variability is kind of similar. So if we get warm in the north, get warm in the south, get cold in the north, get cold in the south. So there's a lot of variation, but we can see the north in the black line. You have the mean anomaly across both sides. On the red you have El Niño, and in the blue I plotted the Antarctic Oscillation Index. And what we can see is that, if anything, it's forcing the temperature in central Chile, which is for where we have the best data, it seems to be very tightly linked to El Niño. Not at all time scales. So for example, in the 2010, I remember arguing with my colleagues from oceanography, like, oh, we're having an El Niño, and I'm like, well, I'm not, maybe you, but in the equator, the characteristics of El Niño, they're not this sort of like canonical El Niño. I didn't put the picture there, but they're very different flavors of El Niño. So how we can use El Niño to predict environmental variability is not so clear cut, except when you have like a clear phenomenon like here and here. Okay. So we have this pattern of remote forcing that will happen all along the coast. In the paper from Chiloé that I showed you a picture before, on 2015, we saw the anomaly going all the way down to Chiloé, which was the first time that it was clearly documented. The other important lesson is that we have this like very clear spatial structure of environmental variability. You can anticipate how sites are going to behave, which is very good from the sense that you can design good experiments because you can know beforehand where you're going to get one or other pattern of variability. But what are the consequences for organisms? That we're gonna hear after me, like Marco is gonna talk a lot about it, Juan Diego too. But I'll show you briefly an example of a paper we wrote, like right around the transition zone, where I showed you have this like very big range of variation. This place, one consequence of this spatially structured environmental variation and the large-scale gradient is that you get breaks in species distribution. You get a process that might be the driver behind the biogeographic structure along the coast. So, for one thing, the area, if you remember, the break takes place here, north or south of here. And the area is very interesting because you get a range overlap of species that they coexist only there. So, for example, in this little square, you get the algae that they coexist with this limpet, and vice versa, like they coexist with this other guy here. They come from the north, they come from the south, and they all live happily together only in these 200 kilometers of coast. It's the only area where you can find them coexisting. So from an ecological point of view, it's very interesting, and we've been like studying that place for a long time. For example, looking at coexistence using the natural experiment, because now we know the pattern of environmental variability. Okay, so on the range overlap area, we have these two limpets that coexist, and the limpets are really closely related. Phylogenetically, they're kind of on the end, their own end branch. However, they have differences to live in the north and in the south. They're not exactly the same. But this is the Phenotypic Plasticity Symposium. So how do these genotypes manage to live at the edge of the range through what their physiology can do? Okay, so this is kind of the same image I showed before, but now we have a site that is further north in Huasco. So Huasco is really already in the northern part of the country, so you have this very smooth annual cycle, very broad, and the sides just south are much colder. But if you look at the blue side, which is the one immediately south, that's the coldest one of all, because it's the one at the core of the upwelling, and it's colder most of the time than the sides that are further south. So you get this like clearly clear spatial structure of environmental variability that is also very predictable. And the species that coexist here, like the northern one, doesn't live. The southern one doesn't live this far north. So if you look using discrete samples, not continuous samples, at the water properties in all these places, you will realize that Talcaruca, which is the coldest one, the one more influenced by upwelling, is the one that gives the lower saturation state of aragorite. Look at it. And the range, it goes below 1, which for some species will be corrosive water. Okay? So the upwelling site is actually a physiologically demanding site. It's predictably demanding. And when you look at what the look at also the VCO2 values there, they're the highest ones also. So the upwelling is having an impact on the intertidal zone, is having a strong impact. And what that means for the species? It means that the black bar is the species coming from the north. This is kind of like an ecological sidelight. Ecologically, the species that is invading the south, because the northern species is been advancing to the south, and through the 20 years of study, we've been seeing it moving south. But the northern species is not contracting the range, it's hanging on there, and the northern species actually, and the southern species, they're doing kind of equally well at the range overlap side. But as soon as you move further south, the southern species is doing very well, but the northern species is decaying. This is the metabolic rate, so it's a measure of how species are performing, how much energy they have to grow, etc. It's a kind of a proxy of the fitness. So what do you see ecologically also is interesting, that the species that is finding its edge of the range towards the equator, okay, so the one that you would expect that it will be extinguishing and pushing back, is actually doing quite well close to the edge of the range, and at the edge of the range is doing just as well as the other one. But also that there's a southern side where both of them are they're under high stress. Okay, so the species that is finding the edge of the range, it has this spatial variation in the performance, but the one coming from the north is just decreasing the performance. Okay, so we have this like spatially structured response of the species. And finally, this is my last slide, and it's kind of a very, very recent stuff that we want to send for submission soon. If you look at demographic rates of all the species across the area we've been studying for too long, the black dots are significant. This is correlation, and in how they have been changing in time this demographic rate. This is the rate of larval arrival. And it's pretty cool that in the north, these are barnacles. Now I'm talking about different species, but the demographic rates of these species in the north, they've been increasing, and in the south they've been decreasing. And this is like has persisted for this like 20-year period. So I'm throwing in there because there's this like slower signal that we don't have enough data to show, but the species seem to be responding also over the long term to this spatially structured pattern of environmental variability. Okay? So that will be great, and thank you very much.
Thank you, Bernardo. Any questions for Bernardo?
Are you monitoring any, but because of the aquaculture, are you monitoring also like a control area where there's no aquaculture or anything like a control?
Very, very good question. We're not because we're putting all our effort in this area because it's a lot of effort actually, And the study that I was showing is what will be a control. Where, for example, where we have welling area. For example, all these sites are non-aquaculture sites. Okay, and they're located in central Chile. What will be very correct from that point of view will be to do something similar, probably around Chiloé area, and Marco maybe will show you some of that. So we're not measuring with the intensity that we're working on aquaculture area, but we're looking at more like basic questions outside aquaculture areas in other places. Okay. Other question? Okay. Sorry.
If you go back to that figure with the temperatures, the 20 years of temperature, with that one. So despite the gaps, all that blue there, I kind of see that on the left-hand side of the figure, there is more heterogeneity than on the right-hand side. That somehow from 2009 onwards, things became more homogeneous along the coast. And I'm wondering if that might have something to do with the instruments, or something happened that made things a little less heterogeneous in space. Do you see that, or am I imagining things?
No, I've looked at it for many hours.
Lack of coffee?
Claro, and lack of coffee will do it. But what we think is that at least this part of the period, these two, it was more, it was a neutral period. And what you're talking about, the instrument, I will agree to be skeptic, because we started throwing away instruments more oftenly. I don't know if after 2008, but now we throw them away like once a year. I mean, we don't let them go for very long. And this data, it should be quality control, like there's some big paper published with it. But I think it might be a bias that we're not looking at here the data from the south, because the north is kind of like better behaved. The sites are more stable than in the south. And also, this is a transitional period, like some weird things were happening oceanographically, at least from 2007 onwards.
Yeah. So I was wondering if this is real, and there's no instrument bias or anything of these things. These sites are so shallow that we could also be seeing effects of changing, wave action or sea breeze dynamics. So things that are happening at higher frequencies that we are not often looking at. So this might be something to think about.
Okay, the last question, so because we are almost at time.
Do you feel that you guessed that maybe the north of Chile is suffering changes in the circulation with more tropical influence patterns, warming more because of getting south? Do you have any information about the oceanography of the regions?
You mean like in the place in north,
Yes.
right? No, not really. Not that we know, because the equatorial influence is really strong, but it's strong through kind of like intraseasonal variation, not so much as in changes of circulation. And we don't have enough data to see that, but we have seen that there's some papers published from around here for Concepcion, where they have longer time here, that it seems that things are changing, but after 2008. After 2008, there's kind of like a step change, which might be related to the general global warming and the expansion of the mid-latitude. Not so much as a tropicalization, because in northern Chile there are really no tropical water. So if you see like the tropical water, it doesn't really reach that far south. And also, and the north has the particular thing that there are absolutely no rivers. So the water property are really related to what's going on in the ocean. I mean, the first rivers are in Peru. I don't know if that answers your question, but I think so.
Okay, thank you, Bernardo. Okay. So next presentation is Dr. Eugenio Carpizo about the phenotypic plasticity of certain muscles in the California current systems. So we are transmitting all the symposium on the Facebook streaming. So maybe we can give the chance if someone connected online wants to ask, to make question, they can do it through the Facebook platform. Okay.
Okay, my first 2 minutes are going to be in Spanish. I asked Sam his permission. He said no. But es mi primera vez en Chile y la verdad es que es muy emocionante para mí por una razón muy personal. Mi profesor de zoología de invertebrados hace 40 años salió de esta universidad. Fue el doctor Alberto Carbacho, que al final terminó aquí en el Museo de Historia Natural, creo. Y él llegó a Ensenada a finales de los 70s, impartió el curso de zoología de invertebrados más inolvidable que se pueda tomar, ¿no? Armó una colección de organismos vivos, organizó un grupo como de 20 personas que se quedaron en vacaciones y fue una experiencia que ya no pude soltar a los invertebrados. Después se me aparecieron delfines y ballenas, pero ya no, eran los invertebrados. Entonces, para mí pues, es un gusto y no podía perder la oportunidad de decirles que el maestro que me dio zoología salió de aquí. Empezando con esto, bueno, mi plática en realidad es como una visión muy personal de cómo nos hemos ido acercando and getting closer and closer to phenotypic plasticity, even personally. You know, I have hair sometimes. My talk is going to be in Spanglish because I'm going to try to use my best English. I'm going to just give you a little introduction about Baja California, and then put in the context of the Anthropocene, and probably most of you know what we are having a lot of trouble in our oceans because of our activities, and then focus a little bit on what is going on in the southern part of the California current system that Bernardo really talked very nice in a way that we're going to put. We have in Baja California, we have like the opposite, but 1 order of magnitude compressed than you have here in Chile, and you will see it. And then I'm going to just give you a snapshots of things that we are starting to do in my laboratory to move into do experiments with animals in the field and in the laboratory with to try to understand better phenotypic plasticity. Well, this is the road to Ensenada. It's a very nice road from basically the border. It goes along the coast, and as you can see, it's a coast full of mountains or next to the ocean. It's very cold, influenced by the California current system, and this is an unusual picture that is very green. Probably it's very, I mean, this last year we had rains that never happened in many years, and in contrast, what you have here in some places, we really don't have rain. We are located out there next to San Diego, and the border is right here. And just to give you an idea, the whole country is like 5,000 kilometers. It's less long than Chile. And the Baja California Peninsula is about 1,500. And this zone is highly influenced by the California current system that goes all the way to here, and sometimes got its influence all the way down here. And well, the main universities along this coast are located in these areas, and we share with CICESE and Ensenada several things. The bay where we are located, and naturally it's getting more important because our mussel aquaculture activities are here in this area, and you will see this story of how we ended culturing mussels. It's very funny because we didn't intend to find Galloprovincialis, and it happens after. This is where the university is located, and as I mentioned to you, this is the Bahía de Todos Santos. That is influenced by the California current here. This is an area full of opaline, and sometimes the bay itself has a lot of influence on this area too. The university is here. This is the city of Ensenada, and it's a very highly complex with a lot of scientists. You know, I think it's one of the cities with the highest proportion of scientists in terms of the size of the city. And I don't know if it's that good or bad, but we're a lot. And we are a wine country too. That's the other thing that I've learned from Alberto. You know, we made a lot of wine, and now it's getting so popular that we have even like a contest every year that people brings their unlabeled wine, and everybody just taste and see who got the best ones. That's the only time that I was able to try to make wine. It was not that bad. I mean, my friend is good to making wine. I'm not. And this is the school that I started. This is where Carpaccio gave me my first lecture in invertebrate zoology, but then in 40 years has changed a lot. And then the message here for me, and I'm keeping repeating it, I mean, we change the environment in a way that we really notice easily in places like that, but not in the ocean. In the ocean it's harder, but we are getting everywhere. You know, like this is in Cancun, in the Caribbean coast, where a friend of mine is working with corals, and she found this motorcycle in the bottom where next to it she has like a culture of. And then we are really changing the ocean in a way that most of you already hear about it. You know, like we thought about this curve, the killing curve. I think it's killing us, no, but it's increasing. And I checked yesterday the level of CO2 in the atmosphere. It keeps rising and rising and rising. No, and seems like the problems are getting worse. And probably most of you already hear about how CO2 works in the ocean. But basically, all the CO2 that is getting to the ocean is changing the chemistry in a way that we never seen for a long time. And this is just a projection of what the pH, you know, it's like 80-50s. And if you, this is based on a work that this international organization on ocean acidification has reproduced. And as you can see, I mean, there's a lot of areas that pH is starting to get levels in the general ocean that they're going to be problematic for the organisms. But the problem for me as a biologist, I always talk with Martin. He's very detailed in the reactions and everything. But for me, it's like, that means that in the ocean, when you have the carbon dioxide dissolving in the ocean, there's a decrease in pH, and then make structures to begin with of the calcium carbonates gets harder. No? But that's one thing in the water column, and the next thing is next to the coast, where we have like a lot of activity, where we have aquaculture activities, we have fisheries around. The problem is a little more complicated, no? And we are starting to see that we need to know the chemical envelope where the animals live. And that's something that everybody's trying to get, because we don't have the sensors, the radar sensors. Martin has been pushing very hard in the north to get like sensors in the water, and he was testing these ones in one of our tanks in the aquaculture facilities, and I went and took a picture with an experiment, but I think now he is out of eyes, I hope. But then it's one of the things that we're missing up there. You know, we still have struggling to have like time series next to where the places the animals are, like the aquaculture or the like the intertidal or subtidal zones. We are getting there. More people is involved in the issue. We started, like Martin and I, probably like almost 10 years ago to start to look things together, and we are still missing a lot of places, as you will see later, where we don't have like a good time series of pH measurement or other things related to the CO2 system next to where the organisms are. Then the first thing that we already know is that every system works very different, and every species and every interaction is going to be more different and more complicated than we thought at the very beginning. That's what it's starting to see, and that's where phenotypic plasticity is starting to get. We know now that every system works different, and the summaries that we made in the past in a very general way is that many organisms are going to be affected, some of them more in different ways, like corals and fish, but still it's like a general thing that we don't really know exactly, because most of those studies were done based on predictions on the open ocean variation. And as Cristian was saying at the beginning of his introduction, we now know that variability in the places where the animals live, it can be very different than what we think in the open ocean. And then we still need to work on that. In the California current system, and I'm going to be talking mainly of the one particular region, it works like this way on a regular basis. No, the influence goes this way. It has some eddies that make some areas more different than the rest. And the Baja California is influenced all the way down to where our state finished. And I will be talking mainly about this region here. This part is about 600 kilometers length. It's 1/10 of the length of Chile. The state this is Baja California, and this is Baja California Sur. La Paz and Los Cabos are down here. And what I'm very interested is that the California current system goes very big on this way. This is the Eugenio, a very interesting place because it has a lot of influence all the way up here. And you still find, like, even, I don't remember if the Kale forest goes all the way down there, but abalones and species like that more common up here are still down here. It's a very isolated place. But what is very interesting to me here is that this part of the coast has like a different oceanographic regime, a little different compared to what we found out here. And the other thing that is very interesting here is that warm waters coming out here, they go into the Vizcaíno Bay, and they influence all this area. And that's where we started a project like many years ago, trying to look for intertidal communities of mussels. We have the California mussel, the Mytilus californianus, and some of the things that I'll be talking to you right next. These are like kind of the models that we use in my laboratory. In my laboratory, it goes from larval biology work to work in the field with sometimes adults, sometimes recruits. I'm gonna talk a little bit first about Mytilus californianus. It's the California mussel. It's very common in our intertidal. It's an engineering species, and I want to talk to you about projects that we did to try to understand how was the distribution affected along that gradient of temperature mainly. Then I want to give you just snapshots about experiments that we have done with sea urchins. Yeah, and lately at the end, I'm going to talk about this species of urchin that is like moving northward and having a an interesting impact in our systems up there. And then just briefly tell you about the possibility of experiments that we can do to know better about what phenotypic plasticity is having on the species there. Well, this is the mussel that we have. It's a pretty big species. It goes all the way from Alaska to Mexico, but in this region, I think it has its southern distribution. And what we did with this survey is that we did a prospection along all these dots along the coast, all the way down to Punta Baja. You can have like access on the main road. The red line is the main road. That's a paved road. But the rest of the trip, you have to go. This area is very non-studied because there's very difficult access. You have to go all this way and then back next to this part of the coast, and then you can go in a little bit, but it's still a little complicated. When we were doing this work, we said that we do unpaved oceanography because the only way to get there was basically on the road. Then we did the initial survey and then choose like 5 sites to come back every season and try to get a better idea of what was going on in that area. That whole area that we sampled is like about 600 kilometers. It's like 1/10 of what Bernardo was describing in his work. In the very north, we divided the state like in about like 300 kilometers in 300 kilometers, and the northern part was more like more population and more accessible, and the southern part is isolated and with less people. Also, the mussel, the Mytilus californianus, is used by people with 2 purposes. It's eaten by like personal consumption, and also is used for in the lobster traps. They use it to attract lobsters to the traps, and then people goes to the intertidal and do that. But beds can be very dense, like in this place, Bajamar. You know, it's a very dense bed. And what is funny here is that that place is protected by a tourist place for play golf, and it's very. I mean, the intertidal is nice. And then we move into other places like the Erendira. It's a place like it's about 70 kilometers south of Ensenada, and people used to harvest mussels very intense. Actually, when we started mussels in Baja California, we started like in 97, 80, and doing the ecology, because the idea was to try to culture these species, but at the end, it never happened. When they put ropes on the water, Galoprovincialis was the seed that attached to them, and that's the way the culture started, you know, like with very practical. And you have places like Punta Baja that it's also very interesting because it has like a regime very similar to more northern parts, and the fauna, it's again, there's like upwelling up there, and you can see the density of mussels is very extended, and also urchins. We found here like 290 urchins per square meter in that area. It's very, very dense. If you keep going south, it's get a little more complicated and interesting because there's less substrate. It's much warmer. I mean, you can have like places like with several degrees higher in that area compared to what you see north. And if you keep going much further, you find like these small patches of mussels, and we were able to identify there like between 5 and 7 different species coexisting in the same place in a very narrow area, comparable to what Bernardo was saying in that area. That we haven't done anything with that yet, but I think those places are very interesting to see how phenotypic plasticity and interaction of species can influence their distribution, no? And with the species closely related. Particularly, there's one species that we found there that is from the Gulf and goes all the way up to there and is coexisting with Mytilus californianus in a way that I don't think is there any other place that happens. And well, with this project, we did like the whole survey, basically based on the model that they developed in Pisco, the group that they work all the way along the California coast. In a more not that intense. They did like 10 transects or 30 transects. I don't remember. We were able to make like 6 in every site with a lot of people involved every time because we have to count a lot of things. This is north. This is south. This is the place that I mentioned to you. The animals are coexisting in a way that doesn't happen anymore. And Mytilus is very abundant in many places. But in particular, I wanted to show you here that in this place, that is very isolated, no way that people goes to collect there. Mytilus was very abundant when we sampled. That was like probably 10 years ago, and it's disappearing now. I mean, we haven't been able to sample again, but it's decreasing in a way that we don't know yet what the causes could be. The sites are really related to each other. You know, north becomes closer to the north, and there's very separate in a way. And that's what they look like if you put them in context with north or south. Northern sites are more like this, you know, with a lot of anemones and sea stars, but also with the wasting of the seas that I don't know you hear about it. There's that they killed a lot of species of echinoderms in California, in Baja California. This species that was very abundant is almost gone on those sites. Again, we were able to find like tens, like 20, 30, 40, like in the order of tens sea stars per square meter to almost nothing. It's really amazing the way that it's affected. But in the south, you find the species that they are not really very common down there. And one of the things that we anecdotically look is like this species that is very common in the south part is getting very common, like 100 kilometers more up. And this is really happening. I mean, just looking at the literature, I saw the paper that was just came out, and there's a lot of species that are changing the range. And because it's becoming more common that we have like these warm temperatures affecting changing the system, we are seeing things that we never have seen it before. I mean, Bernardo was mentioning the El Nino slide that he showed, like where the blob was up there in Northern California, and you see that this means that several species are changing, probably permanently, the distribution of these species. And with this scenario that I'm telling you, I think this is a small laboratory, 600 kilometers long, just to test what you are seeing here in 6,000 kilometers. It's a different scale, compressed, but with a lot of difference in terms of temperature, probably pH that we still don't know. Now I'm just going to give you a small hint of the things that we have been doing in the lab to try to move our experience to try to understand better how phenotypic plasticity is working with these animals. And I'm just going to give you 3 examples of works that we've done with Mytilus, and this is the red orchard that is commercial in Baja California. In the last one is a species that I mentioned to you is like moving north and probably having effects that we still don't know in our fauna, on our local fauna. Well, the work that I've been working with is a student in my lab that did his work with Mytilus californianus. And basically what she did is we tried to do a reciprocal transplant, but we couldn't do it because the logistic was a little hard. And what we decided to do is just move muscles from different places, I think from Punta Baja, Heréndira, and Bajamar to Punta Morro to do like a transplant and see if they behave, how they behave. No? It was a very simple experiment, and I think we're going to do it much better next time. But just to give you an idea, the bay, this is where the experiment was conducted, and the bay has like a pattern that it's in spring and winter and spring, you have like aragonite saturation going up, and then with the blob during the 15th and 60s, it can change very hard. I mean, very, very rapidly. And this is the sites where we got the muscles. In the distance, the maximum distance was like 200 kilometers between Punta Baja and Bajamar. And then this is the conditions, basically same temperature. Punta Baja is very highly influenced by opaline. And we went, collect the muscles, put them in boxes, distribute them, measure them, and distribute them along the intertidal. And what we did is put like 3 replicates for each side, and with holes on the corners, and try to put them in a way that they were submerged basically at the same time. We had like to wait and see what was the line to make it happen. And then also we extracted DNA and made like this very cheap ratio of RNA DNA to try to have a very general estimate of how they were working. And this is like the average size of the animals, like 18 millimeters, 2 centimeters, and that's the way they perform. This is, as you can see here, the ones that are original, and we call it like the control because we didn't move them from their place. They behave a little better than the rest. And in terms of growth, again, they did better than the rest, and they did comparable with Punta Baja. In terms of RNA DNA ratio, we didn't find it really something very interesting, but there seems like a decrease in the ratio when increasing temperature, and probably the muscles, they were really like being moved to a place down up there. No? This is the temperature that the Hubble showed. You know, they were exposed to sometimes to 16, but in that year it was very. This is underwater. It's not. This is when they were underwater only. And pH measuring total scale at very rare occasions it reaches 7.8. No, it was very not. They really were not. There was no acidification problem there. Then we have been done some work with sea urchins, and basically we have been gaining a lot of experience raising urchins. Right now in the lab we are able to raise from fertilization to metamorphosis like 7 different species of urchins that we can work in many different ways to try to test hypotheses related to phenotypic plasticity I think in many of these points of their larval cycle. But in the past, what we have done is experiments to test how the live may behave when they are heat shocked to different temperatures. Now we are doing like the tolerance curve to the whole range, but this is experiment with the rats. As you can see, when you move them very fast to more than 23 or 24, they die basically very fast. And this is an experiment that we did to try to see how that affected metamorphosis. And this we measure like partial metamorphosis and total metamorphosis after being exposed, and trying to acclimate for 24 hours only to 20 and 23 degrees. The larvae were raised at 13, and they reached competency in like 23 days. And what we noticed is that when you increase temperature, incomplete metamorphosis starts to appear very fast. And when you try to acclimate them only for one day. Actually, they die at 29. 29 is just too hot. But what is interesting to me is that we're starting to register next to the coast 26 degrees in the very shallow areas, and then it wouldn't be strange or rare in the future that these larvae are exposing to those high temperatures. And then just to end my talk, I'm going to talk of this species that is starting to appear more often in our cultures. This is Arbaces stellata. It's an orchid that it's a more tropical distribution. It goes, but it has been in our coast, but a very, very small numbers, but it's increasing super fast. And I have my colleagues from the ecology that they go and dive more than I do. They have noticing that the populations are growing very fast. It's very aggressive. And what we did is we documented the cycle, the maturation cycle, and we found that these guys reproduce most of the time. Like 10 out of 12 months, we were able to make them spawn and raise the larvae, all the way through metamorphosis. And one of the things that we noticed already is that compared to urchins like the reds and the purples, these larvae reach competency faster. That we don't know if that can be good or bad. We'll see. But also they support higher temperatures, and they share the habitat with mainly probably the red and the purples. And we think that this is going to become a problem soon for the populations in our sites. And it would be nice to have like a good experiments to see how phenotypic plasticity competing with food or other things can affect these animals. Mainly, and just to finalize, I'm trying to use the experience that I've been reaching in my lab, and with the help of people like Martin in the chemistry, trying to do experiments for all the larval cycle to try to contribute in a parallel way with what phenotypic plasticity can do with the species in this ocean that is changing that much. And just to the end, we were this weekend, as Cristian says, talking about a common garden experiment with Galo provincialis. No, this is a species that we culture in Baja California. And as I mentioned to you before, that species, it was like circumstantial the way that we ended up culturing. But it's going to be very nice to have this opportunity to share and start experiments together and learn and keep learning the way that I started 40 years ago with a Chilean guy again, but much later. Thank you much.
Okay, we have time for a couple of questions before the coffee.
No question.
No question maybe for the coffee?
I have a question, but it's about, like, you have kinds of species, like a lot of different echinoderms, let's say that. But you work a lot also in the larval development. Can you see like some trends in terms of like the tolerances or the plasticity in terms of changes in temperature across the life history?
What we have done before, Juan, is like we have racing and describing just for many of the species. I mean, for the purps and reds, it was done before. But what we are doing now is exactly that, just to see like what we're exposing them to a range of temperatures to do the curve and start from fertilization to the first larval stage for all the species, and then all the way to metamorphosis for all the species, and see if we have like and the other thing that we wanted to do is go for populations that are more south, the warmest ones, and then compare if they have like some difference in the way that they respond, and then try to acclimate to different temperatures. I mean, those are like different ideas to go in that way, and then later on probably do competition experiments.
Thanks.
Okay, thank you, Eugenio. Now we have coffee, and we came back at 11 in 20 minutes max.